Hello, sir. So you are not audible.
सर आवाज नाही येत.
आता येतोय का?
हा येतोय सर.
येतोय ना, बरं बरं बरं. बहुतेक तो माझा हेडफोन व्यवस्थित लागला नसेल. आता येतोय का सर्वांना? यस
हो
स्टुडंट,
येतोय सर.
येतोय. हा
Yes,
बरोबर.
sir.
हा तर आपण अगोदर काय पाहतोय? आपण ओके, इनिशियल पार्ट व्हाट वी हॅव स्टडीड हिअर, वी हॅव स्टडीड that is the cell, I can say anatomy there at such here. Okay, we are studying here the anatomy of our cell only there at such here, how the cell is working. Okay, not working but how the structure of cell is there at such here. Okay, so in first part we have seen there the plasma membrane or the cell membrane there at such here. Now we will today see nucleus. Nucleus is the important part there. Anyone anything knows about nucleus, what the nucleus of cell contains? Yes, come on fast. Yes, what different parts?
Genetic material.
Nucleus contains
Genetic material.
excellent, great. Genetic material. Nucleus contains genetic material. What is the name of that genetic material inside the nucleus? Anyone?
DNA.
DNA.
and RNA.
Yes, great. RNA, DNA. Okay, but that particular DNA or RNA, it is present inside the nucleus. Okay, inside the nucleus. And anything else present there in nucleus? Nucleolus is also there, na? Am I right?
Yes, sir.
Nucleolus. Okay, then one my question is to you there, that DNA is present either inside the nucleolus or somewhere else.
Chromatin.
Okay, chromatin is present where? Either outside the nucleolus or inside the nucleolus.
Outside
the
Outside. Okay, great. Okay, good answer there at such here. Okay, that chromatin is present outside the nucleolus there, or nucleoli there at such here. Inside one nucleus or inside each cell. Okay, per cell generally one nucleus is present there. Okay, only the exception is of some okay, molds. In case of some molds inside what here, one cell, so many nuclei are present there at such here. That is the one exception. And one more exception is there, that is of the algae. Okay, in case of algae also per cell more than one nucleus is present there at such here. And see here, inside one nucleus, so many nucleoli can be present there at such here. Okay, more than one nucleolus can be present there at such here. This is the one thing there, you remember it here. Okay, that is idea here. Then nuclear membrane we are studying there. What here? The nuclear membrane is the outer envelope consisting of a 40 to 70 nanometer wide space there as such here. A 40 to 70 nanometer, what I will say here, wide space. Yes, 40 to 70 nanometer wide space. Okay, it consists of what here? 40 to 70 nanometer wide space. Basically, the space is present in between two layers here. The outer layer of the nuclear membrane is studded with ribosomes here. Outer layer of the nuclear membrane is studded with the ribosomes and is continuous with endoplasmic reticulum. Remember, I will show you the diagram of the nucleus there as such here. Okay, from the anatomy of that particular diagram of the nucleus, everything will be clear to you there as such here. Now, can you see this particular diagram of nucleus? Yes, students, please reply. Can you see?
sir.
Yes, sir.
Yes, that diagram I have taken from internet there as such here, just for your understanding purpose. You can see it here. This is the nuclear envelope here. Okay, nuclear envelope. Okay, nuclear envelope generally made up of the two layers there. Okay, then this is a chromatin. You can see it here, chromatin. Okay, chromatin is present outside the nucleolus. Then this is a nucleolus here. You can see it here. Nucleolus. 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 Okay, then this particular outer thing there as such here, or the outer nuclear envelope, it is attached to the endoplasmic reticulum. You can see it here. This is the endoplasmic reticulum there as such here. Okay, this is what here, the endoplasmic reticulum there as such here. Okay, as well as to the endoplasmic reticulum, the ribosomes are attached there. Can you see the ribosomes here? Yes, you can see it here. Okay, you will be able to see these particular things. That is nothing but the ribosomes there as such here. Okay, this is a ribosome here, ribosomes. Okay, which are attached to the endoplasmic reticulum here. Okay, and these are the nuclear pores. Okay, we just see here the nuclear pores. How the nuclear pores are there as such here, like this. Okay, these are the nuclear pores. Okay, which are having fixed diameter there as such. Yesterday we saw only there as such here. Okay, this is the thing. Okay, then nucleolus and all these things are there as such here. Okay, this is the diagram for the nucleus there as such. Okay, the nucleus of what here? Cell there as such here. The developed form of the cell we can say, that is nothing but what here? Eukaryotic cell only here. In case of prokaryotic cell, nucleus is not present, but the what here, the genetic material or the chromatin, that chromatin is just present in the what here, dispersed form in the cytoplasm of a cell there as such here. Inside the nucleus, nucleoplasm is there. That is nothing but nucleoplasm is the one semi-solid liquid which is present inside the nucleus. The name of that particular nucleus is nothing, that liquid, semi-solid liquid is nothing but it is what here, nucleoplasm there as such here. And inside this particular nucleoplasm, this chromatin is present there as such here, as you can see in the diagram here. Okay, and nucleolus is also suspending there as such here. This is a nucleolus. Nucleolus don't have the membrane there as such here. Outer membrane it don't have there as such. Okay, so that is idea here regarding the part of this particular nucleolus here as such here. Okay, this is idea here. Then after this particular thing here, the two layers of the nuclear membrane at places are fused together, forming circular pores, forming nuclear pores, circular nuclear pores, which are about 50 nanometer in diameter. Okay, the diameter of the nuclear pores, they are circular in nature, nuclear pores, and their diameter is 50 nanometer here. Okay, the nuclear membrane is crossed by several factors which regulate the gene expression and repair the gene damage as soon as it occurs there as such here. That is idea here. Okay, now the next thing is nothing but it is what here, by the nuclear chromatin. Okay, nuclear chromatin, which is in the form of shorter pieces of what here I will say. Okay, short pieces there as such here. Okay, short pieces of thread-like structures called chromosomes, of which there are 23 pairs. 46 chromosomes are there together, measuring about a meter in length in a human diploid cell. Of this, there are 22 pairs, so that is are coming under autosome category, 44 chromosomes, and one pair of sex chromosomes, either XX or XY. XX is present in female or XY is present in male there as such here. The one chromosome is small in case of male. Okay, and the two chromosomes, they are of equal size in case of female there as such here. Each chromosome is composed of two chromatids. Okay, each chromosome is composed of two chromatids connected at the centromere to form X configuration, having variation in location of the centromere. Centromere means what here? This point of attachment. Remember it here, where this is one chromatid and this is another chromatid here. Okay, one line is one chromatid, second line is, okay, that is also another chromatid there. Okay, like this, two chromatids, one chromatid and plus one chromatid, these two chromatids are crossing to each other at one point, and that point of crossing or point of intersection, it is called as centromere. Remember it here, it is called as centromere. In case of different type of chromosomes, this particular, the position of the centromere or this particular point of contact of two chromatids, it is different, and based on that we are identifying or classifying the chromosomes, having variation in location of the centromere there, depending upon the length of chromosomes and centromeric location. Okay, 46 chromosomes are categorized into 7 groups from A to G according to the Denver classification, adopted at a meeting. Okay, adopted at a meeting in Denver, USA there at such here. Okay, don't do the annotations, students. Okay, like this there at such here. Okay, so this is the idea here so far regarding the part there at such here. The slide is over there. We will move to the next slide. Chromosomes are composed of 3 components. Basically, the chromosomes are there. Those are composed of what? 3 components there at such here. Chromosomes are composed of 3 components, each with a distinctive function. Okay, each with yes, chromosomes are composed of 3 components, each with a distinctive function. These are deoxyribonucleic acid, deoxyribonucleic acid, DNA, comprising about 20% or 20%. Then ribonucleic acid, it is RNA, and about 10%. Okay, 10% RNA, 10% DNA, 20% RNA, 10%, and the remaining 70% consist of the nuclear proteins that include a number of basic proteins, histones, neutral proteins. Okay, and acid proteins there as such. Histone is a basic protein. You remember it here. The question I will ask. Okay, remember the histone is acidic, neutral, or basic protein. Then in that condition, you should know that histone is a basic protein there as such here. Okay, the pH of it should be this protein. It should be higher than that of what here? Than that of 7. That's why we are calling to that protein as a basic protein. DNA of the cell is largely contained. DNA of the cell is largely contained in the nucleus. Okay, DNA of the cell is largely contained in the nucleus here. The only other place in the cell that contains small amount of DNA is mitochondria. Nuclear DNA along with histone nuclear proteins form bed-like structures called nucleosomes. Remember it here. Nuclear DNA along with histone nuclear proteins form bed-like or bed-like structures called nucleosomes. Bead म्हणजे गोटी कशी असते? ओके, गोल गोटी त्या ठिकाणी त्या ओके गोटी सारखं त्या गोटीला उंड केलेला असतो ते त्या ठिकाणी लाईक दिस हियर. Nuclear DNA along with histone nuclear protein form bed-like structures called nucleosomes, which are studied along the coils of DNA, which are studied along the coils of DNA. Remember it there as such here. Which are studied along the coils of DNA. Remember it there as such here. Okay. Nuclear DNA carries the genetic information. Nuclear DNA carries the genetic information. Remember it here. Okay. Nuclear DNA there as such here. You remember it here. Okay. Nuclear DNA carries the genetic information that is passed via RNA into the cytoplasm. Okay. Nuclear DNA carries the genetic information that is passed via RNA into the cytoplasm for manufacture of proteins of similar composition there as such here. During cell division, one half of DNA molecule acts as a template. Okay, one half of DNA molecule acts as a template for the manufacture of the other half. For the manufacture of the other half by the enzyme DNA polymerase, so that the genetic characteristics are transmitted to the next progeny, of cells. It is called as replication here. Okay, it is called as replication. This is the basic thing there. During cell division, one half, okay, of DNA molecule acts as a template for the manufacture of the other half by the enzyme DNA polymerase there as such here. Don't strand as the DNA. Okay, these two strands are separating from each other here, and each strand can act as a template. Okay, from one strand its complementary strand is formed, and from another strand its complementary strand is formed. Like this, the two complementary strands, whatever are newly formed, okay, they will form new DNA molecule there as such here, and this particular new DNA molecule will pass to the new cell, whatever will get formed after the cell division here. It is called as mitosis division. Remember it here. It is called as mitosis division. During whatever the cell division is occurring inside our body, it is generally occurring by mitosis. Remember it here. Okay, mitosis is a such type of cell division. Here, the genetic modification is not occurring. Okay, the genetic sequence is as such. The genetic sequence remains constant. Meiosis is a such type of cell division. Here, the recombination of the genetic information may occur there as such here. That's why in case of sex cells, okay, the meiosis type of cell division occurs. Meiosis is nothing but it is the formation of haploid cells from diploid cells here. For your information, I have told this particular thing to you. Okay, so this is idea here so far regarding this part now. Okay, now this diagram already we have seen there as such here. Okay, the DNA means basically you remember it here. Okay, chromosome is made up of what here? 3 parts. What are these 3 parts? Yes, chromosome are composed of 3 parts. One is DNA, second is RNA, and third one is proteins. Okay, DNA is present in 20% amount, RNA is present in 10% amount, and proteins are present in 70% amount. Okay, nucleosomes are there. Nucleosomes are nothing but those are the combinations of what here? Histone proteins and what there? DNA there as such here. Nuclear DNA along with histone nuclear proteins form bead-like structures called nucleosomes, which are studied along the coils of DNA there as such here. Along the coils of DNA. Okay, like this there as such here. Replication we can say here. The DNA molecule. Okay, the DNA molecule there as such here is proposed. The DNA molecule, the structure of that particular DNA molecule, as proposed by Watson and Crick, Okay, in 1953 consists of two complementary polypeptide chains forming a double helical, okay, forming a double helical strand which is wound spirally, which is wound spirally around an axis, okay, which is wound spirally around an axis, okay, around an axis composed of, okay, composed of what here, pentose sugar phosphoric acid chains, okay, it is composed of that particular, okay, what here, it is made up of two complementary polypeptide chains, okay, complementary polypeptide chains forming a double helical strand which is wound spirally around an axis, which is wound spirally around an axis composed of pentose sugar phosphoric acid chains here, okay, and composed of what here, axis, okay, that is composed of pentose sugar and phosphoric acid chains here. The molecule is spirally twisted in a ladder like pattern there as such here. The molecule is spirally twisted there as such here. It is spirally twisted, okay, it is what here, spirally twisted in a ladder like pattern there as such here. Okay, that circular steps are there, okay, circular steps, okay, around one rod, okay, it is like that only there as such here. The molecule is spirally twisted in a ladder like fashion there, or ladder like pattern, the steps of which are composed of four, the steps of which are composed of four nucleotide bases there as such here. The steps, okay, of which are composed of four nucleotide bases, okay, two purines, adenine and guanine, two purines, okay, adenine and guanine, okay, that is A and G, and two pyrimidines, okay, cytosine and thymine, okay, pyrimidine, that is cytosine and thymine, that is C and T. However, A pairs specifically with T while G pairs with C. Okay, the sequence of these nucleotide base pairs in the chain, okay, the sequence of these nucleotide base pairs in the chain determines the information contained in the DNA molecule, okay, or constitutes the genetic code here, constitutes the genetic code here. The sequence of these nucleotide base pairs in the chain, the sequence of these nucleotide base pairs in the chain determines the information contained in the DNA molecule. That sequence only determines the information what is contained inside the DNA molecule there as such, or constitutes the genetic code there as such here, constitutes the genetic code. This, it is called as that information, it is a genetic code. Okay, generally we are calling it as a what here, codon. Remember it here, codon. Okay, that genetic sequence there as such here. In April 2003, sequencing of human genome was completed, which revealed that 23 pairs of chromosomes in the nucleus of each human cell contains, okay, okay, in April 2003, sequencing of human genome was completed, which revealed that 23 pairs of chromosomes in the nucleus of each human cell contains approximately 3 billion base pairs. How many base pairs are there? 3 billion base pairs are there, remember, and each chromosome contains an estimated 30,000 genes. Every chromosome contains an estimated 30,000 genes here in the human genome, in the human genome there as such. Remember it here. Every, okay, and each chromosome contains an estimated 30,000 genes in the human genome there as such here. 30,000 genes in the human genome there as such here. Remember this particular part there as such here. Okay, okay, 30,000 genes in the human genome which carry the instructions for, okay, which carry the instructions for making proteins, which carry the what instructions for making proteins. Remember it here. If we need some additional proteins at genetic level, we can include what here, some sequences, and make our body to synthesize such type of proteins even there as such here. Remember it here. It will be called there as a genetic manipulation here, or the modification at genetic level here. We're getting it here. Okay, this is the thing there. Okay, that is idea here. In the interphase, okay, in the interphase nucleus, okay, in the interphase, okay, interphase is nothing but it is the one phase of cell division there as such here. Prophase, metaphase, anaphase, telophase. Okay, like this. Prophase, metaphase, anaphase, telophase. Interphase. Okay, in the interphase nucleus, that is between mitosis. Okay, that is between mitosis, part of the chromatin that remains relatively inert, okay, metabolically, and appears deeply basophilic due to condensation of chromosomes is called heterochromatin there as such here. In the interphase, okay, in the interphase nucleus, okay, that is between mitosis, part of the chromatin that remains relatively inert, okay, that remains relatively what they are as such here, that remains relatively inert metabolically, okay, that remains relatively inert metabolically and disappears deeply, okay, and appears deeply basophilic due to condensation of chromosomes is called heterochromatin, while the part of chromatin that is lightly stained, that is vesicular due to dispersed chromatin, due to dispersed chromatin is called euchromatin. You remember it here, huh? Euchromatin is what, okay, and heterochromatin is what, this basic difference you remember it here, which remains inert metabolically and appears deeply basophilic due to condensation of chromosomes is called heterochromatin, and which remains lightly stained, okay, in staining process, okay, due to dispersed chromatin is called what here, euchromatin there as such. For example, in lymphocytes there is predominance of heterochromatin. For example, in lymphocytes there is predominance of heterochromatin, okay, while the nucleus of a hepatocyte is mostly, while the nucleus of a hepatocyte is mostly euchromatin there as such here. Nucleus of what, hepatocytes there is the cells inside the liver, those are remaining what, those are basically containing euchromatin there as such here, okay. In the interphase nucleus, okay, between mitosis, part of the chromatin that remains relatively inert metabolically and appears deeply basophilic, that is basophilic due to condensation of the chromosomes is called heterochromatin, and which remains what here, lightly stained, and basically due to the dispersed what I will say chromatin there as such here, dispersed form of the chromatin, then it is also called, it is called as there as euchromatin there as such here. For example, generally in lymphocytes we generally see heterochromatin there as such here, predominance of heterochromatin means large quantity of heterochromatin we see there as such here, okay, and what here, then while the nucleus of a hepatocyte is mostly what there as such here, the nucleus of a hepatocyte is mostly euchromatin there as such here, like this there as such here, okay, that is idea here so far regarding this nucleus part here. Clear nucleus everyone? Yes students, nucleus part, is it clear to you?
Yes.
Yes, okay. Nucleolus. We will see here the nucleolus there as such here. The nucleus, okay, nucleolus. The nucleus may contain one or more rounded bodies called nucleoli. Okay, nucleoli is the plural form of nucleolus here. Remember, nucleoli is what? Anekvachani form. Okay, plural form of nucleolus there. Singular is nucleolus, and plural is nucleoli. Okay, like this here. Like stimulus, stimuli. Okay, like this here. That is idea here. Then nucleolus is the site of synthesis of ribosomal RNA there as such. Nucleolus is the site of synthesis of rRNA, ribosomal RNA, rRNA, tRNA, mRNA. These are the types of RNA you might be knowing there as such. Okay, that rRNA, it is formed inside the nucleolus here. This thing you remember is the important part there. rRNA is formed here inside the cell. rRNA is formed inside the nucleolus. Remember, nucleolus is composed of granules and fibrils representing newly synthesized. Nucleolus is composed of granules and fibrils, okay, representing newly synthesized ribosomal RNA there as such here. Okay, nucleolus is composed of granules, okay, and fibrils representing newly synthesized ribosomal RNA there as such here. Okay, that is the idea here so far regarding this particular part here, nucleolus. This is the structure of DNA there as such here. You can see it here. Okay, can you see it here? Yes, in the structure for DNA, cytosine, guanine, okay, which are attached to each other, okay, by three hydrogen bonds there as such, and adenine and thymine, they are attached to each other by two hydrogen bonds there as such here. Okay, these are the hydrogen bonds. Okay, you can see the hydrogen bonds there as such here. Okay, this is like a ladder. This is like a ladder. You can see it here. Ladder means CD. Okay, steps. These are the steps. On these steps, okay, these hydrogen bonds are forming the steps. Okay, like this there as such here, hydrogen bonds. Remember it here. Okay, like this. Okay, this is idea here. This is a sugar phosphate group. Okay, like this there as such here, thymine, adenine. Okay, like this there as such. Okay, this DNA composes forms what here? Yes, that nucleosomes form what here? That is nothing but the yes, chromatid come and form, yes, two chromatids intersect to each other and form X letter. That X letter is called as chromosome. Means chromosome is nothing but it is formed from DNA there as such here. Remember it here for your information purpose. Okay, this is a very important thing there as such here. DNA. Okay, like this. Okay, then cytosol and organelles are, that is the next part of this particular thing. Okay, cytosol and organelles here. What the thing they have given here? The cytosol or the cytoplasm is the gel-like ground substance in which the organelles, okay, cytosol and organelles, okay, the cytosol or cytoplasm, okay, is the gel-like ground, cytosol or the cytoplasm. It is also called as cytoplasm. Cytosol is the another name for cytoplasm, okay, like this. The cytosol or the cytoplasm is the gel-like ground substance, okay, in which the organelles, meaning little organs of the cells, are suspended there as such here. These organelles are what they are as such here? Yes, these organelles are the site of major enzymatic activities. These organelles are the site of major enzymatic activities of the cell. These organelles are the site of major enzymatic activities of the cell, which are possibly mediated by enzymes in the cytosol there as such here. Okay, enzymes in the cytosol. Okay, basically these organelles are the site of major enzymatic activities of the cell, which are possibly mediated by enzymes in the cytosol. The major organelles are the cytoskeleton, mitochondria, that is mitochondria, okay, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, and the microbodies or peroxisomes there as such here. Peroxisomes there as such here. Peroxisomes. Okay, that is the idea here. Okay, then these are the some idea regarding the parts of that particular cell. Cytoskeleton is there, I told you there yesterday. Okay, three different parts of cytoskeleton are there. That is the microtubule, microfibril, microfibril, intermediate fibrils, and microtubules. These are the three different parts there as such here. Okay, that is the idea here regarding that cytoskeleton part here. Mitochondria, all of we know there as such. Ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, microbodies, or peroxisomes here. Now we will see in short here. Okay, that is the etiology of the cell injury here. Okay, that cell injury we have discussed there as such here. Cell injury, etiology. What is the meaning of etiology of cell etiology there as such here? Yes, etiology means the causes of cell injury, because of which causes the cell injury may occur there as such here. Okay, the cells may be broadly injured by what? Two major ways there as such here. The cells may be broadly injured by two major ways here. The first way, okay, two major ways. They have been categorized into the two things here. The first category is nothing but it is what here? By which different means reasons the cell may get injured. The first one is genetic causes here. The second one is by acquired causes there as such here. Okay, genetic causes and acquired causes. The genetic causes of various diseases are discussed in chapter number 10 in Harsh Mohan there as such. They have told us. But I will discuss with you here. Genetic causes means what here? See, some genes are there which are responsible for certain diseases, like that of rheumatoid arthritis. Suppose we take there. Okay, rheumatoid arthritis. If any one gene is there, okay, inside our body there as such here, which is responsible for that particular disease, rheumatoid arthritis here. If we get that particular disease from our parents, okay, then it is called a genetic cause there as such. If we are getting any disease because of genetic nature here, because of the gene which has come in us from our parents, and that disease has carried from our parents to us. Okay, suppose our parents are suffering from rheumatoid arthritis. Suppose one gene is defective in them, which is responsible for rheumatoid arthritis. Then that defective gene, if it comes in our body, and if we are also suffering from that rheumatoid arthritis, then it is called as the disease of genetic origin there as such, or genetic causes there as such, cell injury. Then in that condition, what will happen here in us also rheumatoid arthritis? Okay, it is autoimmune disease there as such here. Means our body part is specifically, it is okay, by mistake considered as a antigen by our body there, and our body attacks on our own tissues, okay, and causes the damage to our body here. Okay, it is nothing but that autoimmune disease here. It is occurring by genetic causes here. Means in that condition, whatever the damage to our tissue or the cell occurs, it is nothing but cell injury. and its etiology or its cause is nothing but a genetic cause there as such here, like this here. You remember it here, genetic causes here. Okay, second one is nothing but it is what here, etiology of cell injury by acquired causes here. Okay, the genetic causes of various diseases are discussed there as such here. Acquired causes there, the B part is acquired causes there as such. Okay, in chapter number 10, the genetic causes